So thanks everyone for coming. I think I pretty much know everyone here, so you all know how this goes. I'm going to keep it short. My name's Anton, in case any of us have not been formally introduced. So thanks for being here. Quickly thank the library people for letting us host us here. And of course, thank you to Dr. David Noelle for being here. We'll do a quick rundown of some activities coming up. Drinking Skeptically is this Friday in Stockton. Sunday, I believe we have a Turlock brunch. Both of those are very fun events indeed. So go to those. Last night, I guess we had Humans 103. So if you're interested for next month, you can still pick up One Nation Under God. How corporate America created the Christian right, I believe, and join us next month. Also got a book club coming up first Tuesday of August. We're reading Big Science about Ernest Lawrence and the invention of the military-industrial complex. He's the guy that invented the particle accelerator. So that should be a fun one. Other than that, we've got a California Free Thought Day, October 15th in Sacramento. I hear that's fun, though I've never been. But I encourage you all to go anyway, because what David would have wanted. He is not here, by the way.
What do they do at Free Thought Day?
I got a lot of speakers, activities, music. What else? Is that it?
Booths. Other groups in the area.
Yeah.
Hopefully our choir will be together by then.
Yeah, it's a great way to support the local Free Thought community. So go to that. And of course, check Meetup and Facebook for any other activities and goings on. So anyway, also, if you're feeling generous, please support us and drop a buck or two in the basket over on that table. So, I will introduce you now, Dr. David Noelle. Many of you probably know him, associate professor and chair of the Cognitive and Information Sciences at University of Merced, also director of Computational Cognitive Neuroscience Laboratory, and also faculty advisor for the student-run non-religious Atheist and Agnostic United Club. Also a great guy. Might have seen him at some of our events. So I'll let him come up here and give him a big round of applause.
You're a hell of an MC, Anton.
I zoomed in.
Is this working well enough for your turn? Yeah, okay, good. So, hi, I'm Dave. I'm going to talk to you today about how the brain makes ideas, new ideas from old parts. Can people see the slides okay in this lighting? Is this all right? I've spoken to this group before. My talks have kind of taken the form of the neuroscience of something else, where the something else at one point in time was free will, neuroscience of free will, and then I came back a year later and talked about the neuroscience of religious experiences, kind of revelatory experiences. Now, these are both topics that I know something about. I'm a computational neuroscientist, as I'll talk about in a moment, but I really don't generate new research about free will or about religious experiences. That's not what my laboratory does. So these talks were both talks that I just kind of shared what knowledge I have. And I thought it would be fun this time if I talked about something that actually came out of my lab. My lab's the Computational Cognitive Neuroscience Laboratory at the University of California, Merced. And what I'm going to talk to you today is some results that we published in a journal called The Proceedings of the National Academies of Sciences, or the National Academy of Sciences, back in 2013. It was written by my student Trent Creedy, who was the very first person to ever get a Ph.D. in cognitive science from UC Merced. Myself and two of my longtime collaborators, John Cohen at Princeton and Randy O'Reilly in Colorado. So I'm going to tell you a little bit about this in terms of new ideas. But first, I have to tell you a little bit about what I do in my lab, which is computational neuroscience. Computational neuroscience is that branch of the study of the brain that's a little bit like theoretical physics is to the study of physics. So in theoretical physics, you put together these mathematical abstract characterizations of the regularities in the world, try to capture the laws of nature. And then there are other people that do things like build the Large Hadron Collider and go about experimentally seeing whether the theories that were generated actually match up with the real world. There's a lot of work to be done in theory development because theories get complex. You want to make sure that they're internally consistent. You need to have some imagination to figure out new ways that might be, new things that might be true about the universe that people hadn't thought of before. One problem that we face these days in science, however, is that not only in physics, but also in the study of the brain, the study of biology, in virtually every field, you can't describe what we know in just one or two equations. There's just a huge amount of knowledge that we have. So if we want to, say, see what a given theory predicts would happen in a given situation, we don't have enough time to go up to the chalkboard and work out all the math. Instead, what we do is we use computers. We build computer programs that contain all of this mathematical knowledge that we have, and they generate simulations. They say, okay, in this situation, your theory would predict this would happen. And that's what I do. I make models of the brain and try to use them to understand the mind, behavior, cognition. I like to call what I do computational cognitive neuroscience, which stresses that I'm making computer models of the brain, neuroscience, that are trying to explain knowledge and thought and feelings, things that kind of get wrapped up in the term cognition. But other people call the same endeavor other things. Peter Diane likes to call it theoretical neuroscience, like theoretical physics. So there's a variety of labels here, but generally it's building complex theories of how the brain gives rise to all of the wondrous things that it does to our thoughts and our dreams and our desires and our actions. So as we get started here, I need to say a few things about the brain to make sure that we're all on the same page. There's some things that we know in terms of the gross anatomy of the brain, just for some simple examples that I'll be making reference to. The brain is split into two hemispheres, the left and the right hemisphere, and each hemisphere is broken up into 4 lobes. There's the occipital lobe in the back, which is primarily about vision. There's the temporal lobe, which includes auditory areas, but also does a lot of vision work. The parietal lobe that seems to do a lot with space and the like, and the frontal lobe, which includes a section that deals with motor control, but also a whole lot more up here in the front in an area called prefrontal cortex, which I'll be talking about a lot today. In addition to knowing these kind of gross anatomical things about cortex, that kind of crinkly, massive tissue on the outside of your brain, we also know things about the structures that are deep underneath that layer of cells called cortex. The stuff down here is sometimes referred to as the limbic system, and there's a whole bunch of very specialized structures down there in the middle of the brain. Some that I'm going to talk about today include these purple areas here called the basal ganglia, that include things like the striatum, and this thing called the thalamus, which is kind of a relay station. A lot of information goes through the thalamus as it goes from one part of the brain to the other. And we know about these things, and we know how they're connected. There's kind of a language associated with talking about the brain. There's kind of a coordinate system, if you will. So this diagram here is as if I'm facing that way. Anterior or rostral or front refers to where your forehead is. Since there are 2 hemispheres on the left and the right, we talk about lateral being off to the left or off to the right, and medial being close to the middle. The opposite of anterior is posterior, towards the back of your head. And this is going to turn out to be important. We're going to talk about, in certain areas that are more anterior and more posterior, also sometimes called caudal or back. And then the up and down part is superior or dorsal. Think of the dorsal fin on the top of a dolphin. or inferior ventral, like the belly of a lizard. So mainly what I want you to remember here is lateral off to the side, medial towards the middle, and then this anterior posterior front to back difference. Importantly, not only do we know things about the gross anatomy of the brain, but we also know kind of the pieces, the building blocks that it's made out of. One of those primary building blocks is a kind of biological cell called a neuron. And a neuron is a lot like other biological cells in that it has a body and it has a nucleus, it metabolizes, it does the things that cells do. But what's interesting about neurons is they also send signals to each other, signals that can interact in ways that actually allow them to process information. A signal is sent electrically down a long protuberance called an axon, and that electrical signal slides down until it reaches another neuron, at which point, at a junction between the two called the synapse, the electrical change causes the release of some chemical. That chemical is called a neurotransmitter, but it's just some chemical. The other neuron here, the postsynaptic on the receiving side of the synapse, can detect those chemicals, and the detection of the chemicals changes the electrical properties of the receiving cell, thus allowing it to send an electrical signal. Now there are a lot of these in your head. There's about 80 billion. That almost runs up to the number of stars in the Milky Way galaxy. So every one of us have a huge, huge number of these cells in our heads. And they're very densely connected. Each one of these cells could receive inputs from 1,000 or 10,000 or sometimes more other cells. And it's that confluence, that collection of information, signals from a variety of other cells that allow these neurons to calculate interesting things, like bring in bits of light from all over the place coming into your eyes in order to figure out what an object is that you're looking at, say. Now, importantly, we understand these cells well enough to write equations that describe how they work. We can mathematically describe how that electrical signal moves down the axon. We can mathematically describe how that electrical signal gives a release of neurotransmitter. We can mathematically describe how that binds on the receiving cell and how that changes the electrical properties of the receiving cell. So we have all of this detailed knowledge about how these neurons work. Interestingly, we also know, in a lot of detail, how the brain at this level rewires itself in the face of experience. When we have experience, the connection between these cells actually changes, and it happens at the synapse. These synapses can grow, they can die, they can become stronger, they can become weaker. And we have some pretty good characterizations of how it is that experience, that is to say things that cause these cells to get electrically active, change the strength of these synapses, effectively rewiring your brain. So we could, if we wanted to, write a little computer program that simulates one of these neurons, or two of these neurons, or 100, or 1,000, or 10,000. And, well, we don't have computers that can quite do 80 billion yet. We can do some fairly large circuits. Yeah. Oh, didn't know what this meant. Okay. So this is what I use. I take these building blocks and build models of certain neural circuits, certain parts of the brain, and I have some hypothesis. I say, I think that it's this part of the brain that gives us the capability to remember our mother's face. And then I see whether that circuit can actually remember a picture of a face. And if it can, then that's some evidence that at least that circuit is capable of doing that. And it makes predictions about what those cells should be doing when a face is presented that can then be tested by an experimentalist. So I wire them together, make these models here of the hippocampus, deep in the brain, and see what they do, and see if I can use them to help explain how we can do all the wonderful cognitive things that we can do. And one of those wonderful things is to hold in mind a completely new idea. Now, there's two senses of a new idea. There's a new idea that no one on the planet has ever thought of before, no one in history. And then there's a sense of a new idea to you. This is the first time you've entertained this idea. Now, I'm not going to try to explain the whole process of how new ideas arise in our culture or arise in our society, but I'm going to focus on a new idea for you. How it is that your brain can actually hold in mind something that's completely novel. Maybe you thought of it, maybe your creativity gave rise to it, or maybe someone just told it to you. Someone said, here's a new idea. And I'm wondering, what is it about the brain that allows us to so flexibly hold on to these completely different ideas, things that are completely different than what we've seen before? Now, this is an important first step for understanding things like creativity. We can't understand how our brains come up with new ideas until we know how the brain can hold on to new ideas. And so, eventually this might lead to studying things like creativity in an individual. But for now, I'm going to focus just on how can people even think about a new idea. Now, there's a lot of reasons beyond creativity that we might be interested in this. For example, there are times where very smart people say that they can't hold on to a new idea. So for example, you have people like Lord Kelvin here that at one point in time said, I can't imagine how light could simply be electromagnetic radiation. Which of course it is. But he said, that idea doesn't make sense to me. I can't entertain this idea that light is merely electromagnetic radiation. There's got to be some lightness to it. There was a problem in entertaining this novel notion, even with a very bright gentleman. And there are other examples, of course. Hans here was a vitalist who said, I can't imagine how life could simply be a collection of chemical and physical processes. There has to be a spark of life somewhere in there that goes away when you die. And of course there isn't. We know very well that the way that living things are different from non-living things are all in the details of the functioning of certain chemical and physical processes. Some other examples that you might be more familiar with are things like, I can't imagine how the diversity of life could arise without an intelligent designer. And of course, this is again a kind of failure to entertain a new idea. Now, as this audience most likely knows, these inabilities to imagine are not very good arguments against the ideas that are unimagined. As a matter of fact, there's a logical fallacy named after that called argument from ignorance or argument to ignorance. It even has a fancy Latin name. And it says that just because you can't imagine this being true doesn't mean that it's not true. As a matter of fact, the fact that you can't imagine that something is true probably says more about your imagination than it says about the nature of the world. Right? So, if we could understand the properties of the brain that allow us to entertain these novel ideas, or when we fail to entertain these new ideas, we might gain some insights into these situations where people feel compelled by this notion of being unable to hold a new idea in mind. This goes on. These kinds of arguments continue. For example, in my world of neuroscience, there are people that say, I can't imagine how my conscious experience could arise from the biological and chemical processes of a brain. I mean, how is it that all of my loves and pains and hopes, those seem fundamentally different than chemicals and electrical signals? I just can't see how consciousness could be something that the brain does. So this continues to be an issue. But it's really an issue beyond just intellectual endeavors trying to understand what the world is like. Every one of us has to deal with new ideas all the time. We live in a world that's constantly changing. We get a new version of the operating system on our phone, and we need to entertain new ways to do things. There are social changes that we need to deal with. We get a new boss, we have to figure out what's going on. There are just new things coming up all the time, and we need some way to be able to keep those in mind and use those new ideas. So how do we do that? Well, one clue comes from the fact that if an idea is too new, if it's just radically new in every way, we almost always have a terrible time holding in mind. It doesn't make any sense to us if every part of it is new. So instead, a lot of new ideas take familiar things and put them together in new ways. We take old parts and reassemble them in a new way to make a new idea. So we're familiar with the parts, but somehow our brain is able to assemble these things together into a new idea. And when I think about this idea, I mostly think of my nephew Jacob. My nephew Jacob, for a long time, whenever I'd see him, first thing he'd say is, "Can I show you something?" And I know what he wanted to show me. He'd want to show me the latest army of creatures that he had created that were invariably at war with each other. But you can see in his creations that there's like some things that are like snakes, and there's certainly eyes of different kinds, and there's tongues and feet of different kinds, and they're just recombined in ways that you don't see in nature. Indeed, mythological beasts have done this for a long time. The chimera here, again, is a bunch of pieces of animals that do exist, reassembled to make something completely new. So maybe the place to start to think about this is how novel concepts, new ideas, might be formed from the composition of familiar ideas, of familiar parts. There's a lot of productivity to doing this. There are lots of ways that we can come up with taking things that are familiar to us and putting them together in a way that is really useful. Just using physical things in a different way can allow us to be more productive, or can allow us to, do things better in some way. So it's really useful for us, even on a daily basis, to be able to take old ideas that we're familiar with and put them together in a new way. But these new combinations of ideas, they don't always have to be physical things. It's not that we're putting physical things together. We can take economic ideas, take parts of economic ideas and kind of combine them in a different way in order to come up with a new way to help small farms, or to help poor people stay alive, or even social or political ideas we can combine in new ways to make new ways to move forward as a society. So again, the question is, how is it that this thing in our skull, this couple-pound lump of tissue called the brain, how is it able to take these parts and hold them in mind together as a new idea? Now, just to ask that question might seem really kind of strange. It's strange because we're really good at taking things apart and putting them back together again. Ever since we were kids, we took blocks and assembled them in new ways. Even when we learned language, we learned that language was composed of parts, words, and phrases that we could take apart and put together in some new way to produce a new idea. So we have lots and lots of practice as human beings, both physically and in language and in other domains, taking things apart and putting those parts back together. So we might think, well, this is easy. Of course, there's got to be an easy way in which the brain does this. But this is a little deceptive. The things that we find easy because our brains have devoted a lot of resources to make them easy. That over the course of evolution, our brains have developed circuits that make these things automatic and very straightforward for us to do. So it may seem like it's an easy thing, but that doesn't mean that when we get down to actual biological processes doing them, that it's necessarily going to be a very simple mechanism. A good example of this is vision. Just looking around and seeing things seems so easy, and yet a very large fraction of your brain, probably close to a third of your brain, is involved in some way in visual perception. So how could this happen? Well, if you have a background in computer science, if you've done anything with computers, if you've ever used a computer with a von Neumann architecture, which is pretty much every computer that's ever been made. This also seems like an easy problem, because computers have memories, these things that can retain little bits of zeros and ones, and blocks of those zeros and ones have addresses, like address 1, address 2, address 3, address 4. And if we want to store an idea, we just allocate some of those blocks to the idea. So say we have an idea that's made of a subject, a verb, and an object. We say, okay, address 5 is going to be for our subject, address 6 is for our verb, address 7 for our object, and we fill in a pattern of zeros and ones that correspond to the parts that we want to combine together to make this old idea. And then, if we want to make a new idea, we simply allocate some more parts of this computer memory and fill in the same patterns of zeros and ones, except perhaps in a different order, in a different place with regard to what the subject, verb, and object is. If you know even more about computer science, about computer programming, you'll know that we even don't have to do this. We don't have to have copies of the 0 and 1 pattern here for billionaires. We can actually put those patterns in one place in memory. And then when we want to make an old idea or a new idea, rather than list the pattern for billionaires or foreigners, we just indicate where else in memory we will find the corresponding part. So, for example, the subject of the new idea here says, look in location 13, and you'll find the thing that is the subject of this new idea. This is called indirection in computer science. More informally, computer scientists talk about these as pointers. So it seems easy. It seems natural for us to be able to recombine things. Computer scientists would tell us, hey, it's easy. Just, allocate more memory, have pointers to things in a different order. But the brain isn't like a von Neumann computer. It doesn't have a big block of memory of zeros and ones that can do whatever we tell them to, can just encode foreigners in any part. It's more like an analog circuit, like an old transistor radio. So it doesn't make sense to say, okay, well, I'll store the frequency in the power supply, and I'll ask the tuner to provide power to the system. No, the circuits each do a very specific thing, and we can't just treat them as the same and allocate them to do the same sort of job of holding on to some information. And indeed, if somehow we forced them to do that, we'd end up breaking those very circuits. We know that the brain is like this, that neural circuits are specialized for certain tasks. For example, in the sensory system, we know that a lot of the neurons here in the back of your head are detecting low-level features of vision, where things are bright and dark, and where there's a contrast between bright and dark. We know that primary auditory cortex is around here, that information from the chemical receptors in your nose are around here, and the olfactory bulb. Your sensitivity to touch is detected by neurons in this strip called the somatosensory strip. And here is lonely old taste, pretty close to smell since they interact so much. These cells, they respond to specific kinds of senses, to a touch at a particular place on our skin, to a soft touch or a hard touch. Different neurons will respond to that, or a painful pinch, or something that's hot, or something that's cold. Different neurons specialize in detecting, responding, doing a very specific thing. Another example of this is if we look at the broader visual system, there's something that's called the what pathway and the where pathway. As we go from primary visual cortex down into the temporal lobe, what we find is more and more neurons capture features that are important for recognizing what an object is. Like, is that a face that I'm looking at? Whereas if we go dorsally up into the parietal lobe, we find cells that respond to how things are moving or where they're located relative to our bodies, things that are important for where things are or how I might manipulate them. So even though the location of an object and its identity are kind of bound together in the real world, different circuits in the brain are capturing those different aspects of reality. The different circuits are specializing. There's a fun example of this kind of specialization brought to the extreme. This was a paper published some years ago in the laboratory of Christoph Koch. He had a patient that was having brain surgery, had to have his skull open, and he sunk an electrode down into these cells in the middle of the brain, and he found a cell that when this gentleman was presented with a picture of the actress Halle Berry, this cell got very electrically active. Cool. But even if Halle Berry's face was kind of covered up, it still would get very active. As a matter of fact, the cell would get very active for Halle Berry's name. There was a cell in this gentleman's brain that had specialized in detecting Halle Berry. It was that specific. So again, given that our brains are full of these cells that are very specific, how is it that they can recombine things? That one moment they're involved in thinking foreigners as a subject, and later they're thinking of it as an object. We can't just imagine changing the synapses, the connections, rewiring the brain very quickly for these new ideas, because if we did, we'd kind of be breaking these circuits that have taken years to develop and even longer to evolve their basic structure. And we don't want to do that. We don't want to have the experience that when we meet a new person and see a new face and want to remember the name that goes with that face, that it kind of changes all of the connections so that we can no longer recognize our mother. We don't want to break the things that are already there. So how can the brain possibly do this? It looks like it might have a lot to do with special cells that are in the front of your brain, in the frontal cortex. This area is called prefrontal cortex. It's kind of even more in front than the areas that do motor control, move your skeletal muscles. There are a number of different parts of prefrontal cortex. We're going to be looking mostly at this dorsolateral patch of prefrontal cortex over here. Now, this tissue seems to be really important for holding things in mind. If you get damage to this area of the brain, you have trouble holding things in mind. If various chemicals that help keep this part of the brain working are not in place, as there's a problem like this in schizophrenia. People with schizophrenia sometimes have difficulty staying focused on the tasks that they're working on. So kind of holding things in mind in what's called working memory seems to be an important role for these neurons. But there's still the question of how they can hold on to completely new and novel things. One interesting insight that gave us some intuition as to what might be going on was some work that was done in David Lewis's lab in Pittsburgh, where they found that if you look very carefully at the cells in prefrontal cortex, they form these little patches or stripes where within a patch, the neurons are very strongly connected with each other, even more strongly than other places in the brain. But they don't talk very much with the neurons outside of the patch nearby. Now, if you simulate this, what you can see is that this is a perfect configuration for the cells within a patch to hold on to one bit of information, say the color of something I'm looking at, and a different stripe, a different patch, to hold on to some other bit of information, like its shape. And now if I want to think about a blue circle and now think about a blue square, I just have to change the pattern of electrical activity in the shape stripe and keep the one that's encoding the color the same. I've broken up what I'm thinking about into parts, where the parts are now being encoded in the electrical activity across a group of neurons in one of these little stripes. That's about 1 millimeter there, so they're really tiny little stripes in prefrontal cortex. So that starts to give us a hint. There's this special circuitry in prefrontal cortex that seems to break things up into parts. And if you look at the connectivity between those parts of prefrontal cortex and things deep in the brain, you find these loops. They send activity down to this part in the limbic system called the basal ganglia, through the pallidum, the globus pallidus, through this relay station-like area called the thalamus, and then back up again the cortex. And these loops are things that we've known about for a long time. You can find them anatomically. They were broadly reported in this paper from Peter Strick's lab back in the 80s. It's hard to read. That's a photocopy from the original paper. Here's something that's a little bit easier to see. But what you see is that there are these loops that go from dorsolateral prefrontal cortex down to the basal ganglia, in this case part of the striatum, through the thalamus and back up again. So there's these interesting loops, kind of sending signals down and back up again. And what we found is that the striatum parts of these loops, the basal ganglia parts of these loops, kind of act as gatekeepers. So up here in cortex, like in prefrontal cortex here, the cortex might be coming up with all sorts of ideas. Maybe I should go to the movies. No, maybe I should study. And down in the basal ganglia is where some system learns to decide which of those things to focus on. And it sends a signal through the thalamus back up saying, okay, you guys shut up. This is the one that we should be focusing on right now. So it seems really important for figuring out what we're going to hold in mind. These circuits in the basal ganglia, in places like the striatum, seem to learn from experience, hey, this is what we need to focus on right now. We kind of know how this works. We know that these neurons here, they're labeled here for pyramidal cells. The shapes of the neurons look like little pyramids. Within a prefrontal cortex stripe, they have these dense connections that can keep them actively holding on to some information. But down here in the basal ganglia, through the thalamus, there are signals that can essentially say, okay, stop thinking about that, or no, really stay focused on that and ignore any distractions. And we've made models of this, how learning systems based on a chemical called dopamine can learn from reward when to focus on some things and not focus on others. We can learn that if we stay focused on our studies, then we'll get a good grade. That focusing on certain things will lead to reward in the future. And we've built some models that we published over the years describing exactly how this learning works. And it's kind of culminated in this model called the prefrontal cortex basal ganglia working memory model, which essentially, as I say, has this loop through the striatum, back up through the thalamus that tells prefrontal cortex what to remember and what it can safely forget at a given moment. One thing that's been added to this model since then is the idea that the basal ganglia not only says, okay, remember this, or don't bother remembering this, but it also has a signal that tells the brain when it's okay to let the rest of the brain know what you're holding on to. So there are signals coming from down here in the basal ganglia that just say, hold on to this in this maintenance place in prefrontal cortex. But there are other signals that say, okay, let other parts of the brain know what it is that you're holding on to. We call this an output gate. It's like opening a gate on a gated enclosure and letting the rest of the brain kind of get the information that's in that enclosure. So we have all these ideas, and we pulled them together and said, okay, how could this allow us to think of ideas that have multiple types? Well, maybe there are these stripes of neurons, and maybe there's a stripe for, say, the subject part of an idea, another stripe for the verb part of an idea, another stripe for the object part of an idea. This is an oversimplification, but just as an illustration. And if we want to entertain a particular idea, like doctors hate diseases, all we have to do is we have to make, we need to have enough experience in our childhood development to learn a pattern of electrical activity amongst these neurons that encode doctors, a pattern of electrical activity across these neurons that encode hate, and a pattern of electrical activity across these neurons that encode diseases. And that's great. We now have doctors hate diseases encoded in those stripes. The problem comes when we start to change things around. What if we have something like diseases hate medication, and we've never, ever, ever, ever in our lives ever entertained an idea where diseases was a subject? Diseases aren't something that can do things. They're not a subject. So if we've never, ever, ever seen diseases as a subject, then we'll have never learned a pattern across these neurons that could encode diseases. These neurons know how to encode diseases, but these ones don't. So this kind of a system would have a problem. It wouldn't be able to entertain the idea that diseases hate medication because it's never learned a way to encode in these neurons this notion of diseases, even though other parts of the brain can. So how can this be fixed? It has to do with those loops. So Peter Strick, again, not only found these loops between the midbrain and the frontal cortex, so they go down and then back up again, but he also found that there was this kind of asymmetry to these loops. That not only do these projections through the thalamus come back up to about the same part of prefrontal cortex, but they also kind of loop a little bit more posterior, a little bit further back. And those cells loop a little bit further back, kind of like a spring or a spiral. It's possible for the electrical activity in the cells that are closer to the front of your head to have an influence on what gets remembered or what gets let out to the rest of your brain in areas a little bit further back, but not vice versa. There's an asymmetry here. The anterior part can influence the more posterior parts, but not vice versa. And Peter Strick saw this, mostly in rats. We've since then looked very carefully at the anatomy of various mammals and found this asymmetry to be there in the anatomy. We've used brain scanning techniques that can follow major tracts of neurons, tracts of those axons, and found that asymmetry in human brains, where again, near the anterior point here, there's more of a tendency to connect further back, but not in the opposite direction. This data coming from a brain imaging study done in the laboratory of Walt Schneider with the help of David Bader. So we know that they're there, but we don't know what they do. David Bader wrote this cool review article in 2008 where he said, you know, people that do these brain scanning subjects, they've noticed that different parts of prefrontal cortex get really active when people are doing different things, when you ask them to do different tasks. But they've come up with all sorts of different explanations about what those different parts of prefrontal cortex do. Some have said, well, you know, way out here, this is kind of like your abstract plan. I'm going to go to Vegas. And then back here is something in between, like I'm going to drive to the airport. And a little bit further back is, oh, I have to make a left-hand turn at this light. So there's kind of this abstraction hierarchy across what we're keeping in mind, what we're focusing on, what we need to do. Other people, when they've asked people to do other tasks, have said, nonsense. This has to do with how complicated the various components of the thing that you're thinking about are related. If they're related in a really complicated way, then they involve the anterior parts, and if they're related in a simpler way, then it's further back. Or it has to do with the various ways in which we control our behavior, or how we deal with conflict between the choices that are presented between us. There are all these explanations for different tasks. And nobody had an idea how to sort this out until we came along. This is Trent Creedy, my student, Randy O'Reilly in Colorado, John Cohen in Princeton. We got together and we thought, okay, what happens if you just build the circuit? If you just had a brain that had little stripes of prefrontal cortex neurons that were connected in this asymmetric way through the basal ganglia and the thalamus, and you let the synapses develop using the same mathematical rules of synaptic change, synaptic rewiring that we know exist, to various kinds of rewarding experiences. Remember this complex concept. Remember that doctors hate diseases. Remember this other thing, in such a way that you can repeat back to me what was the subject of that. Oh, it was doctors. And see how the synapses change, how the brain rewires itself in order to do something different in the anterior parts of the brain versus the more posterior parts. So that's what we did. We built this really complicated computational model that had stripes and had a basal ganglia and had a thalamus. This complicated model, what we found is it did something really kind of interesting. So we had some stripes that were anterior, closer to the front. And just for simplicity, I'm just going to have one layer, so one collection of stripes a little bit more posterior, a little bit closer to the back. And the front stripes were associated with different components of an idea, like subject, verb, and object. But when presented with something like medication hates diseases, it didn't store in the pattern of firing of these cells medication and these hate and these diseases. Instead, the code for medication was stored in one of the back stripes, the code for hate in a different back stripe, and the code for diseases in another. And what was encoded in the frontal stripes was essentially what part of the back stripes were holding the subject, the verb, and the object. So the connections from these neurons down to the basal ganglia that control these neurons say, hey, when you're looking for the subject, open up the output gates here, and you'll find a pattern of activity that corresponds to diseases. So here, diseases is the subject. So this is diseases, hate, medication. Diseases, hate, object, medication. Now in this case, these neurons never ever had to encode diseases. They just had to encode look in this stripe, which they could have done for other sorts of ideas. And we just had to have seen diseases at some point in time, that there's some stripe that can encode them. And suddenly we have a system. And this system developed this on its own. We did not make it do this. We just let the brain, the model brain, rewire itself the way it needed to in order to remember these things. And this is what it came up with. So this is the diagram that we had in the paper. This is a little bit backwards from the last diagram, in that the front is on the right and the back is on the left. But again, the idea is that if I want to know the object or the patient here, it encodes a pattern of activity that points to the basal ganglia for the corresponding stripe in the more posterior area that says what the patient is. In this case, steak, and says, yeah, let steak out when I'm being asked about the patient of the concept that I'm holding on to. We wanted to compare this model with other modified models to see what were the properties of our model that really made this work. Would other models do the same thing? So we took our model and we ripped out the output gating. So now the rest of the brain can always see what's in all the PFC stripes. It doesn't wait for the basal ganglia to say, okay, show the rest of the brain what your electrical activity is. We tried just a model that didn't have anterior and posterior parts. It just had one big collection of stripes. This was the original prefrontal cortex basal ganglia working memory model. And we even went so far as to just take kind of a glom of neurons and say, hey, learn it on your own. Just learn to store whatever you need to store in the electrical activity of these neurons and see whether it can do the task. Now the next slide looks a little bit complicated, but I'll explain it very quickly. We wanted to have different difficulties of new ideas. So we had different tests that we gave these networks. The standard test involved making sure that every slot, like the subject, verb, or patient, had seen every possible filler, every possible word. So this is kind of the easy case. Every slot had seen every possible filler, so it would be easy to generalize to recombinations of those. And we tested some random recombinations and looked to see how well it did. Then we had what we call the anti-correlation case. So the anti-correlation case involved having the network during its entire period of developmental learning, its entire childhood. It never saw Bob do anything with soup. So these things were perfectly anti-correlated. Bob and soup just do not appear in the same room. And then we tested to see whether it could entertain the notion of Bob eating soup. Or did it learn that this is an impossibility, this is something that it can't imagine? And we had even the harder test of, I've never seen a given filler in a given role. I've never seen Bob as the patient. Bob is always the actor. Can it entertain an idea where Bob is the patient? And these are the three categories: the standard, easy generalization test, anti-correlation test, and the test where I've never seen Bob as a patient before. I've never seen this filler in this role. And the different colored bars are showing the four models that we compared. Our full indirection model that contained everything we knew about this anatomy, the one that just had output gating, the basic PBWM model, and the simple recurrent network that had to learn everything on its own. And what's on the y-axis here is the accuracy on a percent scale of the models on being able to entertain the new ideas. And what you see is that kind of learning everything on your own doesn't do so well. The gray bars are kind of low, which means that having an evolutionary history is a good thing. There's some structure to our brain that's there to start, and it isn't all learned during a period of development. It's good to have the stripes there, some structure. And the models did pretty good on both the standard case and the output gating model. Actually did really well even on the anti-correlation case. It could entertain the idea of Bob eating soup even though it had never seen Bob in the same room with soup before. But when we went to the point where I had never seen Bob as a subject, or I'd never seen diseases as a subject, only the full model was able to do very well. All the others, well, frankly, they sucked. So this gave us a real hint. It said, well, maybe for some kinds of novel ideas, ones that are only slightly novel, you don't need all of these mechanisms. You could actually have a brain that maybe didn't have as much prefrontal cortex, didn't have all those spirally connections, and still get by. But if you want to really entertain a very novel idea where I've never seen this used in this way before, all of these mechanisms are important to be there in order to be able to entertain that new idea.
What does generative mean in that sense?
The idea is that what has been learned for representations of ideas here, it can essentially generate any possible combination of things that you could imagine. Generative is a word that's often used in language to describe how I can produce a sentence that's completely novel, and there's this huge, almost, people describe it as infinite, but at least it's a very, very large range of sentences I can generate. Similarly here,
roughly the same as creative, then.
Yeah, in a way, it's dealing with a novel situation, it's representing a novel situation, but it's doing so in this kind of combinatoric way where the space of possibilities is just huge that could actually be generated. Whereas the number of cases where you obey some anti-correlation, there's a much smaller set of things that are novel ideas of that kind. Does that make sense?
Yes.
We struggled with that name a lot. We kept trying to think of a different word, but it turned out that it was the best thing for the literature because of this reference to linguistics. So this is what we found. We found that our model, if it just learned the way that we know that the brain rewires itself from experience, getting reward if it does things right, it generated these representations that don't encode things in the world, but encode other parts of the brain. So we have neurons now that are not encoding just Holly Berry, or encoding redness, or the location of an object, but they're encoding that part of the brain. That part is important. Remember, that part is what we need to use. Now, if you think about it, that's kind of like what the computer scientists call pointers. Here we have something that's not referring to billionaires or foreigners or loves, it's referring to another memory location. So it's as if the brain naturally learns to produce something like pointers. But it's important to note that there's a difference. In computer science, I can put any address in here that I want, any memory address. It can be perfectly generative in the sense that it can point to any memory address it wants. But in our model, it's very sensitive to the developmental experiences that it has while it's rewiring itself. And so if the model does not get adequate practice recombining things in different ways, it will not be able to combine things as flexibly as if it has had a lot of experience combining things in different ways. So this suggests that this is why, if we have a child that has a very poor developmental environment, doesn't get a chance to try new things out, that they're really stifled later in dealing with new things that are being introduced to them. Perhaps one of the reasons for that is during that early period of development, they did not learn this structure, this ability to point to other parts of the brain quite as robustly. And maybe this is true for all of us. Maybe there are some things that we just never saw in our development that makes it harder for us to entertain a certain new idea. Certainly there are some combinations, like a healthy argument, that we can entertain easier than others, like what's healthy abuse? I don't know what healthy abuse is. Maybe if we try really hard, we can imagine what that is. So there are limitations in our development that affect where we can have a pointer and where we can't. So it isn't just like computer science. It's something that's really sensitive to the learning that happens while we're children. So I think that this is a really interesting first step towards trying to understand creativity. It allows us to understand how the brain in the frontal lobes is holding on to completely novel ideas that it has never seen before. And now the question becomes, where do those novel ideas come from? In our case, they were all just told to the network, here's a new idea, remember it. But is there some way that these neurons could just kind of randomly fire in a new way, and then some other part of the brain, maybe somewhere in the limbic system where there are circuits that are involved with reward, could say, hey, did that work? Is that rewarding? And come up with new combinations on its own. Now, we haven't simulated that yet, but this seems to be a step in the direction of understanding how these processes of creatively trying new combinations might arise. And that's what I got for you. And I hope that we have some time to address your questions.
So when you were talking about the dopamine, sort of reinforcing decisions and rewriting a little bit, does that play into determinism and free will to some extent? When you're thinking if determinism is true, how can people be accountable for their actions, that sort of thing? Anything else?
So there's a philosophical stance, as you may know, in the world of free will called libertarianism. It has nothing to do with the political position. It essentially says that for a choice to be free, it has to not be caused by anything else. No other cause could have caused that choice, because then it wasn't free. It was caused by that other thing. But as early as people like David Hume, around the time that this country was formed, philosophers have said, that's wrong. We don't want our choices to not be caused by things, because that's just randomness. We want our choices to be caused by our knowledge and by our past experience and by our character and so on. So what it means to make an intelligent free choice has to include some sort of causes of things about our knowledge and our experience, which are encoded in our brain. And our past experiences of what was rewarding and what wasn't rewarding, which is something that dopamine helps us learn. So whether you want to call that free or not might be a matter of philosophical contention, but it's certainly consistent with this notion of what some philosophers call compatibilism, that there is a notion of free choice that still can arise from a materialistic mechanism. The dopamine story, if I can just do an aside here, is actually another cool example of what I do. So, long time ago, there were these computer scientists that said, we want to make programs that can learn from reward. How can we do that? And they came up with these algorithms, and one of the key parts of their algorithms was a little subsystem that would predict how good it is to be in a particular situation. Oh, this looks like a situation where I'm going to get reward in the future. I like being here, or this is a bad situation. And a key variable was the difference between those two after taking an action. So if I'm in a situation where things look great and I take an action and now they don't look so great, maybe I was too optimistic a moment before and I learned something. Or if it looked really awful and I take an action and things look better, then maybe I was too pessimistic and I learned something that helps me choose actions. Years later, a gentleman named Schultz in Germany was recording from these dopamine neurons in the midbrain. And he found that when you give a monkey a reward, like a sip of juice, these cells fire. They're happy. So people thought, hey, dopamine is about happiness and rewarding good things. But if you always shine a light a second and a half before the food, after a while, they fire for the light. Well, that makes sense. The light is now associated with the food. It's a rewarding thing. But they stop firing for the food. The monkey still wants the food, but now they're firing for the light and not the food. And if you're mean and you withhold the food, then they fire for the light and then they dip. They get really quiet when the food would normally be delivered. What is this? This is the difference between expected reward before, when the light came on, and after the time that reward would be delivered. In the case where the monkey doesn't know the association between a light and a reward, the reward is a surprise. Oh, life sucks, I'm strapped in a chair, I'm doing a stupid experiment. Oh, food! Positive going change in reward. When it learns the association with the light comes on. Oh, I'm getting rewarded. And when the food comes, there's no change. I knew I was going to get rewarded. The light came on. And when the food is withheld, there's a negative going change. I thought I was going to get rewarded, and I wasn't. And some people at the Salk Institute, when I was in graduate school in San Diego, said, that's exactly this mechanism in that machine learning algorithm that people came up with before. This difference in predicting reward. And they came up with computational models of how these circuits in the midbrain can learn to choose actions based on reward, based on the merger of these two lines of work. And to date, those models have not been taken down after many, many challenges. Though a student of mine who's defending in a few weeks might knock it down a little bit. So this is a good example of how these models are useful. And it's also an interesting example of how some chemical like dopamine might change over the course of scientific history, exactly how we're interpreting what it is. I walked all over a bunch of other questions like that. Yeah.
I know neurons are the star of the show, but do you guys mathematize the limbic system? And what kind of information would the limbic system carry?
So usually the term limbic system involves this big area of the brain. So it includes neurons and all the things that those neurons do. But I imagine that you're pointing at a bunch of chemical things that go on both there and elsewhere in the brain as well. Things like hormones being widely dispersed, or even neurotransmitters not just showing up at a synapse, a connection between two neurons, but neurotransmitters that are kind of widely dispersed over an area, which does happen. I mean, there are hormones that are released that cause us to change our mood or things like that. And there are models that make use of those things to the degree that we understand them. They tend to be more difficult to test because they tend to be things that kind of have this slow, mushy time course, so we can't bring somebody in a lab and, wait for them to have a good mood and then see what they do or something like that. But you can get people in a good mood by giving them M&Ms. It turns out that you can get... So there are models that include things like hormones and other chemical processes. They just don't seem to be the star of the show in terms of what's going on kind of a second-by-second level of cognition. Yeah.
I have kind of maybe two questions or topics that are kind of different. One is, kind of from the brain, I've also heard that the body's nervous system is kind of almost like a brain as well, and it connects to the heart. And for example, there's stories or books that have been written about people that have had heart transplants, and they take a lot of characteristics of the person who, whose heart they transplanted or something like that, and it seems like something to that. Have you heard anything along that line?
Yeah, so there's a couple of things to say here. One is there's kind of an adage in the world of skepticism, which is before you try to come up with an explanation for a phenomena, make sure that there's a phenomenon to explain. And so if someone comes to you and says, no, really, I saw a ghost upstairs. How would you explain that? How would you explain that the ghost just walked through the wall? How could it? You first have to find out what they really saw, right? There's no data that when you give somebody someone else's heart, it changes their personality in a way that's consistent with the other person's heart. There's just no data at all that happens. But that doesn't mean that your body isn't important. Here's an example, and it has to do with dopamine. If I take a rat, and I put the rat in a tub of water, it's distressed. It doesn't like to swim. It wants to get its feet down. And it'll swim and swim and swim, and eventually it'll say, I give up, and it'll just go into a dead float. If I pump its stomach full of probiotics, it swims longer.
Probiotics meaning bacteria?
Yeah, various kinds of bacteria that are useful for the digestive process. If I pump it full of probiotics, but I sever the vagus nerve that connects the gut to the brain, it goes back to swimming for a short time before giving up. And it turns out that the greatest amount of dopamine in your body is in your gut, not in your brain. So maybe part of what keeps us seeking reward is the dopamine makes the bacteria in our gut happy. So it's not even our other organs, it's another creature altogether. Makes them happy, and they send a signal through sensors in the gut up the vagus nerve to the brain saying, hey, things are good. Right? That's wild. We are more than our brains. We're more than our bodies. We're connected to all of these other things, and it determines our behavior.
One other quick question. I saw this article in New Atlas. It's called Mind Reading Technology Can Now Decode Complex Thoughts. I don't know if you've heard of that, where they're able to kind of like you're saying, finding pieces of the brain and looking at brain scans in kind of real time, and they're able to now scan at a resolution where they can see sentences like being formed. Like you thought this sentence, which is kind of...
Yeah, so the trick here, it has to do with this generativity thing. It's really great if you restrict the range of things that people are thinking about. But if you let it completely open, it doesn't do so well. But this is a good example of why there are theoretical neuroscientists and experimental neuroscientists. Because for the longest time in the world of experimental brain scanning, the kind of analysis you'd see would be things like, let's show someone a face and see what part of the brain is most active. Which, you know, it's cool, but it doesn't tell you very much about how faces are recognized. And only recently, by having various kind of theoretical techniques and more complicated computational and mathematical techniques, have we started to tease apart the whole circuits in the brain that are involved with different processes. And we've gotten to the point where we can kind of look at all the activity in a brain scan and say, hey, that's more consistent with the last time that someone was thinking this word, as compared to thinking this other word for a small set of words. And it's cool because it isn't even based on the senses. There's a phenomenon called binocular rivalry. So if I put in one eye a picture of a house and in another eye a picture of a face, your brain doesn't like that. Your brain says, no, I'm looking at one thing. I can't see face house. So you have this experience of focusing your attention first on the house, so you see the house for a while, and then you see the face for a while, and then you see the house for a while. So your consciousness is kind of drifting back and forth between the two images. Now the images haven't changed. What's coming into your eyes stays exactly the same, but what inside you're aware of is changing. And we can detect when you're seeing the house and when you're seeing the face. So we can tell by what neurons are active in your brain whether you are aware of the face coming in your eyes, which is always there, or you're aware of the house coming into your eyes. That's cool. And kind of scary.
That's how I see it.
I'm just curious, when you showed the last graph, we all know people that are just kind of weirdly creative. They just come up with things always, and it's always that person in the group, you know, like try and you're just not that creative, right? Do their brains function with more open gates? Is it a structural thing? They have different chemicals? What's going on there?
So the short answer is we don't know. There's a lot of stuff that we don't know about creativity. But the vast majority of theories of creativity have this kind of generate and test flavor. That is to say, there's a process that more or less randomly tries something out, combines things together, and then there's another system that analyzes that and says, is that a useful thing to do? Should I really do that painting? Should I really, write that poem? No, that's stupid. Or, yeah, sure, I'll give it a try. So the question then becomes whether the more creative person is just kind of more loose in trying different things, that they just kind of pop up more frequently and they're trying a whole bunch more things in a period of time, or whether they have just super well-refined judgment systems for saying, that combination, I can make that work. That's a good one. And it could be a combination of those things. It could be that, to some degree, creativity involves letting go of the constraints, letting more different things combine and think about them rapidly. But it also it requires kind of knowledge, skill, a refined sensibility of what combinations meet the demands of whatever it is that you're trying to do. Does that help?
Yeah.
I've always understood the hippocampus to be the center of formation of memories. Would that mean that there are more circuits that are leading to the hippocampus than other parts of the brain?
So the hippocampus is this kind of loopy shaped structure that's in the midbrain. It gets its name because somebody thought it looked like a seahorse, and hippocampus is what, Greek, I think, for seahorse. And it's one of these parts of the brain that's connected to everything. All these parts of cortex send inputs into entorhinal cortex, which goes into the hippocampus. And from entorhinal cortex, there are connections that go out everywhere in the brain. So here's an interesting opportunity. If I want to remember an event, like looking at your face now in the evening, with a little bit of sweat on my brow, a little bit out of breath, all of those sensory experiences are going on in different parts of my brain. Sensing my lungs, sensing the heat, and all those signals are going to the hippocampus. Now the hippocampus has a chance then to try to put those things together. But it needs to put them together in a way different than what I've been talking about here, more than just entertain them for a short period of time. I want to remember this moment tomorrow. So the hippocampus is one of the places where we get new neurons even as adults. There are new neurons that come up from stem cells in the hippocampus. And what appears to happen is that thing about rewiring the brain is different in the hippocampus, where it does rewire things really fast. That a few neurons, like the Halle Berry neuron, can very quickly say, okay, put together the heat and this face and this time of day and being out of breath. Change all those synapses to pull all those things together into the connection of this very small set of neurons. And the hippocampus is very special. At any one time, only a very small set of neurons get to be very strongly active, and the other ones are quiet. So there's only like a few that get this rapid rewiring to capture this moment. So yeah, it seems to be really important for memories, particularly these memories of episodes, memories that involve binding things together.
Yeah, so if we want to talk about personal identity and what makes you you, which is essentially what you remember about your life and all your relationships, I would think that the hippocampus is more the essential you than any other part of your system.
So this is a really deep question, right? So the philosopher Dan Dennett, who I might have mentioned before, he describes the self, who you are, as the center of narrative gravity. Now, what does he mean by that? The center of gravity of an object is not a physical thing. It's a point in the object where mass is equally distributed around that point. So it's not a thing in reality. It's a mathematical notion. It's a property of the object. So what Dan says is you have all these memories of all these events, and where they all intersect, that's that point, that's you. You're the main character in all of them. And that's what he calls the self. But it's tricky, because if you take somebody that has had the hippocampus removed, it's not like they don't have a sense of self. You can have a short conversation with them. They seem to have personality traits. But then 5 minutes later, they don't remember what happened. So is the memory of your history you? Or is there something more to you, like your tendency to be nice or your tendency to get angry? It's very tricky to define what the self is. Maybe it has something to do with bacteria in your gut.
How concerned are you about the advances in artificial intelligence?
So I worked for an artificial intelligence company before I went to graduate school, and I still do some machine learning work now and again. And let me phrase it this way. I was at a meeting in Philadelphia last summer where I talked to one of my colleagues, a student of John Cohen's, a guy named Matt Botvinnik, and Matt said to me, I'm going to leave my lab in Princeton, and I'm going to go work for DeepMind, this company that Google bought that's doing all this great machine learning work. And you know why I'm doing it? Because what they're doing is exactly the same as what we were doing in 1985 when we were making these brain models. That is to say, this big resurgence in artificial intelligence is using almost no new techniques at all. What it's using is the huge number of computers that Google and Facebook have, and the huge amount of data that they have access to, like every YouTube video that was ever made. So they have more experience to give these machine learning algorithms. And still, with all that data, what do they learn? They learn that YouTube videos have cat faces in them. They learn to recognize what's a face and what's not a face. They, by far, have not gotten to things like, I want power, or other kinds of abstract concepts. So we're going to see some really great advances. We're going to get to see things that take sensor data and tell you whether you're going to have a heart attack soon, or that can drive your car for you, and all those kinds of things. But in my lifetime, I'm not going to have to worry about these things causing problems for our society. Another good example, if you watch, there are videos online on YouTube where you can see the latest DARPA challenge of humanoid robots. And the humanoid robots were given the task of walking up to a door and opening the door. And these videos show the robots walk up to the door, not find the doorknob, and then fall over. So if you want to protect against artificial intelligence, close the door. We're not there yet.
I just have one more real quick one. So, you said you create programs that simulate. How on earth would you have the knowledge and researchability and code? You could not possibly. This is a purely practical question. You couldn't possibly have time to actually code the software, right?
So yes and no. So one of the cool things about computer programs is I can write a little subroutine once, and then it can be called all sorts of times, right? So there's a lot of reuse. So if I write out the equations once for how that electrical signal goes down an axon, all of a sudden every single neuron that I model has that part. Right? Now, if I wanted to kind of put in every connection from every neuron to the other for 80 billion neurons, no, I don't have time to do that. So instead, not only do we have smaller circuits, but we depend on those circuits being shaped through a developmental process, through these other mechanisms of strengthening and weakening of synaptic connections. So those things, they just emerge like machine learning results in the models. So some of it comes about from the modeling process. And the parts that don't, it's a matter of just the magic of software that we can reuse the bits of knowledge that we have over and over again. Yeah.
Have you been, I'm fascinated with this, have you been able to model any actual organisms and then satisfactorily predict what they're going to do based on your model?
So there's a lot of models that can predict actions in certain constrained domains. You can do a lot in like classical conditioning domains. There's actually some really cool models of addiction and addiction recovery. I have a friend that did some models on dyslexia and essentially was able to predict what sorts of remediation techniques will help children with dyslexia more than other remediation techniques using models of this kind. So predicting the behavior of organisms, both humans and other kinds of organisms, within some constrained domain is something that's done all the time. It's needed to be done in order to test to see whether the models are real, whether they're matching something in the world. But to get a whole organism, everything about an organism, that we're just starting to get to. There's projects to try to model everything about a worm. Trying to get the whole nervous system, all of the ways that it interacts with the environment, and they're going to see. can we capture everything that's relevant about that one? And then they'll move on to larger organisms, probably something like an insect like a fly or something like that, and move up and up and up. Did that answer your question?
Yeah, yeah. I'd like to have a worm.
You know, what you're going to find is you're going to find that there are programs that are in no way intelligent, but they're cute and fuzzy and I like them, so I treat them as if they're intelligent, right? So you're going to find that people start attributing smarts to things that don't have a lot of smarts before you actually have programs that are very complicated. You can already see this in there. What is it called? Takeuchi or something. There was this thing years ago where you had like a little key fob with a little digital animal that you had to feed and things like that. Oh, I gotta feed my animal. Very simple little program.
I understand you gave a talk on free will at an earlier time. Do you have a recommendation for a book since I missed your talk? Where can I get the cognitive neuroscience perspective on choice? And while euphemistically, well, I just think free will is silly because obviously we don't control our heredity. We don't control our environment. The inputs are not something that we choose, yet we seem to think we have some control over the output. And you must have some references that you would recommend for that.
Yeah, so there are books across the whole spectrum. There's one by Peter Tsao, T-S-A-O, that tries to find some way to give freedom to a notion of neural systems having free choice. But when you get right down to it, it comes back to Dr. Dennett. Dan Dennett has spent his lifetime thinking about these questions, and as early as Elbow Room, and up to I think the most recent one was called Freedom Evolves, that talks about these things. He has thought about these things so thoroughly, and I think has probably the best answers, even though he doesn't always drill right down to the specific neural circuits. The basic ideas are strongest with him. You can pick up things from other people too. There's like essays by people like Pat Churchland and Paul Churchland that talk more about the brain systems involved. And
I just want to point out, our Stanislaus Humanist has 2 YouTube channels, and I'm busy transferring the videos from one to the other.
Yeah.
But if you go to YouTube and do Stanislaus Humanist David Noelle, you'll find his first talk.
Well, really? So it's online?
yeah, there is a version of the free will talk that's online where you get to see me waving my arms in the air.
And there's a basket over here for your donations to make it all possible.
Which you're free to do.
Yeah.
Oh, you get the love basket.
I'm sorry, what?
You get the love basket.
Did I get one?
I don't know.
It's a free love basket.
What does to do with it?
Anything else that people want to talk about?
Last one.
Yeah, just kind of for fun question. I know within the last few years they've gotten biological neurons to talk to computer circuits, and you have computer circuits that mimic the brain. Are we anywhere near being able to augment, just plug in new stripes of neurons and plug in new ideas?
So most of the work here in trying to have an interface between biology and electrical circuits is in the world of artificial prosthetics. So if someone's lost their arm, you want to give them the ability with their brain to control a robotic arm. And they've been able to do some really remarkable things with this. They've not only made actual prosthetics for humans that are better, but they can do things like send it over the internet. They can record the motor commands from a monkey in one place over the net and have it control a robot arm in another place. And it all works seamlessly. Again, it depends on this learning component, though, because everybody's brain is a little bit different. So you can't interpret the signals exactly the same. You have to have a system that kind of learns, okay, when they're moving their arm this way, this is the signal that I'm seeing there. And those work. Memories, we're not quite there yet. Part of it is I can see where your arm is and then say, okay, that's the pattern of activity. I can't say, what are you remembering right now, and really trust what you're saying. So it's hard to do that kind of learning thing for things like memory. But if you ask me, just having Google over here is already a kind of augmentation, and I don't mind that it's interfaced through these neural circuits rather than directly from these.
All right, let's give David a hand.
Thank you very much for coming out.